Hi everyone, today we're going to be solving AQA GCSE Biology Higher Tier Paper 2H. Charles Darwin proposed the theory of natural selection. Many people at the time did not accept his theory. There was a different theory at the same time as the Darwin's theory. The different theory said that changes in an organism during its life could be inherited. Who proposed this theory? The theory was proposed by John Baptist Lamarck. You just have to write Lamarck for the answer. Studying fossils helps scientists understand how living things have evolved. Figure 1 shows a fossilized snake. Explain how fossils in Figure 1 may have formed. So the dead snake was covered in sediment and the soft tissues decays, but the bone doesn't decay. Over the course of time, the bone gets replaced by minerals. There are many types of rat snake in the world. Table 1 shows the type of rat snake. You can see a Japanese rat snake, which is green and grass. The type of environment that it lives is in grass, and the color of the snake is green. And then there is Texas rat snake, which is pale brown in color, and also the type of environment that it lives is dry and dusty. The different type of rat snake have evolved from similar ancestor. The rat snake have evolved to suit their environments. Explain how the Japanese rat snake evolved to be different from the Texas rat snake. When talk about evolve, we will take into consideration for natural variation within a particular population where some individuals are better adapted and the better adapted individuals are more likely to survive and reproduce. Since this particular answer wants a level 2 mark, so it has to be detailed with coherent logical links. So the example answer can be written as follows. Initially, there were lots of different colors of rat snakes. These colors are determined by their genes. Some of these colors are closer to the green of the Japanese grass environment. So the green colored snakes were better camouflaged. So the green snakes were more likely to survive and reproduce. So the surviving offspring will then inherit these genes, which is going to give them a green color. Many species of snake become extinct. Give one reason why species might become extinct. When we are talking about extinct, we have to understand extinct means mass death. Often this can be due to new diseases or any catastrophic event like drought or eruption of volcanoes, okay, or new predator arriving in a particular area or a sudden changes in the surrounding which the animal was not able to adapt to. So there can be many possible answers. I'll just mention few. You just choose any one of them and write in the answer. It will always be correct. A gardener wants to add compost to the soil to increase the yield of strawberries. The gardener wants to make his own compost. An airtight compost heap causes anaerobic decay. Explain why the gardener might be against producing compost using this method. Anaerobic decay often produces a greenhouse gas called methane, which can also contribute to global warming. So we will say methane is produced, which is a greenhouse gas, causes global warming. Whenever giving an answer that says explain, we must have the statement plus reasoning. So in this case, the statement is anaerobic decay produces methane, and then the explanation is, reasoning is, which is a greenhouse gas. The gardener finds this research on the internet. A carbon to nitrogen ratio 25:1 will produce fertile compost. Type of material to compost are chicken manure, horse manure, and pig moss. When looking into the combination, the carbon to nitrogen ratio in chicken manure is only 7:1. In horse manure, 20:1, and the pig moss is given here, but X we have to find out. So to find out the ratio or carbon to nitrogen, we will do 9.8 over 0.2, which gives us 49. That means the ratio is 49:1. Since 49:1 is much higher, so the closest to 25:1 we can see is this one. Which type of material in table 2 would be best for the gardener to use to make his compost? Justify your answer. So we can see from the table, horse manure is the closest to 25:1. So the answer will be horse manure because carbon to nitrogen ratio is closest to 25:1. Some of the leaves from the gardener's strawberry plant die. The dead leaves fall off the strawberry plant onto the ground. The carbon in the dead leaves is recycled through the carbon cycle. Explain how carbon is recycled into the growth of new leaves. So the carbon in the dead leaf is recycled. And then we have to say how the carbon is recycled into the growth of the new leaves. We really have to be very specific to the given example. So how to start about it? First of all, we have the leaf, which is decomposing. The leaf will produce CO2, and a leaf which is doing photosynthesis will take in the CO2. And that CO2 will be used for producing glucose, and then the leaf will die. To get a 6 mark here, we will have to have a detailed coherent. So our answer should be detailed plus coherent plus there must be logical links. So an example answer can be written as follows. Carbon compounds in the dead leaves are broken down by microorganisms. They respire, which releases CO2 into the atmosphere. The plants then take up the CO2, which they use in the process of photosynthesis and produces and converts the carbon dioxide into glucose. The glucose produced is then used to make proteins, which are essential for the growth of the new leaves. Figure 2 shows 2 strawberries. Both strawberries were picked from the same strawberry plant. Both strawberries were picked 3 days ago. The strawberries were stored in different conditions. Strawberry A has mold growing on it, and strawberry B is fresh. So in terms of understanding, first of all, when we look into strawberry A, we would say that there is mold. So the mold requires microorganism, and the microorganism also requires warmth and oxygen. Along with it, it requires moisture. Give 3 possible reasons that may have caused strawberry A to decay. So we would say strawberry A is stored at a higher temperature, or it is stored in conditions with more oxygen. Also, we can say strawberry A is stored in place with more moisture. And one more added point that you may also include is that the storage condition where strawberry A was stored had more microorganism, which is responsible for the decay. Many different types of animals are produced using selective breeding. Some cats are selectively bred so that they do not cause allergies in people. Suggest 2 other reasons why people might selectively breed cat. When we are asking the 2 other reasons, there can be 2 reasons that the 2 general distinguishing point that can come into mind is that first of all, to improve the quality of the life of the cat so that the cat does not get diseases and stuff like that. The other one is to improve the cat as a pet so that it is not like aggressive, all right? It is more docile. The points we can write are to eliminate specific genetic defect or for aesthetic reason, such as to make them less aggressive or more docile or look more beautiful. Selective breeding could cause problems of inbreeding in cats. Describe one problem inbreeding causes. Inbreeding can cause a particular organism to pass on recessive disorder, or the cats that are produced by inbreeding can be more susceptible to diseases. Many people have breathing problems because they are allergic to cats. The allergy is caused by a chemical called FeLD1. Different cat produces different amounts of FeLD1. A cat has been bred so that it does not produce FeLD1. The cat does not cause an allergic reaction. Explain how the cat has been produced using selective breeding. This particular answer requires a level 2 response. So it has to be detailed, coherent, and there must be logical links. So how to go about this particular breeding? First of all, we will have to choose a cat for the purpose that it has the least amount of FeLD1. Then we'll breed, and we choose the offspring with the least amount of FeLD1 and then breed them back again. So the answer can be written in this way. The cat with the desired characteristics, the lowest FeLD1, are allowed to breed. Their offspring will produce a variety of levels of FeLD1. But those with the lowest FeLD1 levels are then allowed to rebreed again. This is then repeated over many generations so that over time the cats produce less FeLD1 or no FeLD1 at all. A student plans an investigation using mold. Mold spores are hazardous. Give one safety precaution the student should take when using this investigation. Because mold spores, they can fly. So it's very important. They can mix in air and they can basically get into our breathing system and be inhaled. So wear a mask. A student made the following hypothesis about the growth of the mold. The higher the temperature, the faster the growth of the mold. The student planned to measure the amount of mold growing on bread. The student used the following materials and equipment: slices of bread, sealable plastic bags, a knife, a chopping board, and mold spores. So basically, the idea is that to have the bread and the mold spores inside a plastic bag. Describe how the materials and the equipment could be used to test the hypothesis. We must have a level 2 answer for this one, so it has to be detailed, coherent, and logical. And the result should be quantitative, and the method should be repeatable. So in terms of giving details, the answer can be written as follows. Take 4 slices of bread and cut them into squares of side length 2 cm. Add 6 pores to each square. Place them in each plastic bag and place each plastic bag at different temperature. Example 20, 30, 40 and 50 degrees Celsius. Then leave for 4 days. Measure the percentage cover of mold on the pieces of bread after 4 days. So whenever we are mentioning that we are using 4 slices of bread, this is the part where we are making it detailed. Each length 2 cm, meaning that this is controlled. It was placed in for 4 days, also controlled variable. The percentage cover, this is our dependent variable. The mentioning of 20, 30, 40 and 50 degrees Celsius, this is our independent variable. Since this particular answer is limited to the number of marks that it's going to provide, so we did not mention how we are going to keep them at 20, 30, 40 or 50 degrees Celsius. Give one variable the student should control in the investigation. So we already talked about it. Amount of mold on each bread piece or the type of mold that is used must be same for all bread piece, or the amount of air in the plastic bag because molds require oxygen or respiration, or the size of the pieces of the bread, or the type of the bread, or the amount of moisture within the bread. Any one of these could be the answer. Another student did a similar investigation. Figure 3 shows the results. We can see day from 0 to day 10, the amount of mold in units. At 42 degrees Celsius, the amount in mold in units is increasing much faster, and it's going to a much higher amount compared to that of 14 degrees Celsius. Determine the rate of mold growth at 42 degrees Celsius between day 2 and day 7. So day 2 and then day 7. We would have to extrapolate. 56, day 2, 4. So 56 minus 4 divided by 7 minus 2. This gives us 10.4. So it grows 10.4 units per day on average. The growth of mold shows decomposition of the bread. Give a conclusion about decomposition from the results in figure 3. So we can say at 42 degrees Celsius, the bread decomposes faster. So the conclusion will be decomposition occurs at a faster rate where the temperature is higher. So the relationship can be described in this way. As the temperature increases, the amount in mold also increases. So, increased temperature means faster rate of decomposition. Figure 4 shows a reflex in the iris of the human eye in response to change in light levels. We can see the pupil is very small here. So, the radial muscles are relaxed and circular muscles are contracted. The pupil is smaller. When looking into the pupil, the iris have circular muscle and radial muscles. So when the pupil is smaller, the radial muscles are relaxed and circular muscles are contracted. Is the opposite when the pupil is larger. So radial muscles will be contracted and circular muscles will be relaxed. Muscles always work in antagonistic pair. Muscles are only able to contract. So that's why only the contraction is shown in the diagram. Describe the changes in the pupil and the iris going from A to B in figure 4. Explain how these changes occur. Refer the changes in light level in your answer. Since the question has 4 marks, we have to describe 2 marks. And for those same points, we'll have to explain 2 marks. So description plus explanation. We can say the pupils at B are dilated. So it is in low light levels. The explanation will be the radial muscles are contract and circular muscle relaxed. Some people wear glasses to improve their vision. Figure 5 shows light entering the eye in a person with blurred vision. Figure 6 shows this condition is corrected with glasses. We can see the blurred vision is occurring because the light is after the focal point, and with lens we can see the light is directly into the fovea. Explain how the blurred vision is corrected. So the first option, in the first one we can see the light does not focus on retina. So this is myopia, nearsighted. With the help of the lens, the light is then directly focusing on the retina in a single spot. So clear image. The question says explain how the blurred vision is corrected. So we will give the answer in this way. Figure 5 shows myopia where light focuses before the retina. In figure 6, the lens bends the light so that it focuses onto the retina. 2 student investigated reflex action times. This is the method used. Student A sits with her elbow resting on the edge of a table. Student B holds a ruler with the bottom of the ruler level with the thumb of student A. Student B drops the ruler. Student A catches the ruler and records the distance as shown in figure 7. Step 1 to 4 were then repeated. So in this diagram, we can see at the point of 10, the student has caught the 10 centimeter, the student has caught the ruler. So just two ways the student could improve the method to make sure the test would give a valid result. So looking back into the steps, we can see student B drops the ruler. Since the student B drops the ruler, it must be dropped from the same height every time. Since it is dropped, it must be dropped without any force. So dropped with no force every time. The student have to catch it. So the student A must catch with the same hand. Since a ruler is used, every experiment must use the same ruler because different ruler might have different mass, which can affect the distance that is dropped by the ruler. When talking about valid result, we must think that this is a repeatable or controlled variable to produce valid result. So the points that we can write is that drop from same height, use the same type of ruler. Table 3 shows student A's result. We can see total number of tests done is 7. What is the median result? So the median refers to the middle of the ordered list. So to find the median, we have to put this data from the smallest to the largest. The smallest one is 106, 106, then 117, then 115. Then 117, then 120, 123, and 125. Since there is a total number of 7 tests, the 7 divided by 2 is 3.5. That means the fourth term will be the median. Is the middle of the order or median. So this is the fourth term from both directions. So 117 will be the answer. The mean distance the ruler was dropped is 116 millimeter. Calculate the mean reaction time. So reaction time in second is root over mean drop distance in centimeter over 490. 10 millimeter is equals to 1 centimeter. So since we have a 116 millimeter drop, so we will do 116 divided by 10, which gives us 11.6 centimeter. So reaction time in seconds, we will find out square root of 11.6 over 490. This gives us 0.1539. Since our answer only wants up until 3 significant figure, so we will write down 0.154. The students then measured student A's reaction time using a computer program. This is the method that is used. The computer shows a red box at the start. As soon as the box turns green, the student has to press a key on the keyboard as fast as possible. The test is repeated 5 times and a mean reaction time is displayed. Student A's mean reaction time was 110 milliseconds. Using a computer program to measure the reaction time is likely to be more valid than the method using a dropped ruler. Give 2 reasons why. So mainly when a computer is used, the human errors are removed. So the 2 points that we can write, there is no indication beforehand when the color will change in the computer screen. And for the number 2, we can say the measurement of time is more precise when doing it with a computer reading than from a ruler. And finally, also we can say the resolution of the computer timer is much higher. One of the points that can be added along with this particular points is that when a particular person is about to release the ruler, the other person might be able to detect when the person is about to release the ruler. So that can affect the reaction time. A woman has a head injury. Her symptoms include finding it difficult to name familiar objects, not being able to remember recent events. Suggest which part of her brain has been damaged. The part of the brain that is responsible for finding familiar object and remembering recent event is cerebral cortex. Now, let's look into a particular brain and understand the functions and the portions that we need to know and their functions. So this particular part is the outer layer, which is the cerebral cortex. It is there for memory, intelligence, language, and etc. This is the pituitary gland. This is the hypothalamus, which is there for regulating temperature and water levels. This is the medulla that controls the unconscious activity, such as breathing and heartbeat. This region is the cerebellum that coordinates movement and keeps balance. A man has a head injury. He staggers and sways as he walks. Suggest which part of his brain has been damaged. Since the person is unable to keep balance, cannot control movement, so that is cerebellum. Figure 8 shows an image of a small section of DNA. Figure 9 shows the structure of small section of DNA. The question asks, what is part B? The part B is looking into a phosphate molecule. So we'll just say phosphate group. This one is named deoxyribose in DNA. Generally, typically, we'll just have to remember as the name sugar. This entire portion is a nucleotide. In figure 8, the structure of DNA shows 4 different bases. There are 4 different bases and they always pair up with the same pairs. Which bases pair up together? Adenine always pairs up with thymine and cytosine pairs up with guanine. Or we can write A with T and C with G. Syndrome H is inherited condition. People with syndrome H do not produce the enzyme IDUA. Figure 9 shows part of the gene coding for the enzyme IDUA. So there are strand K from a person with the syndrome H, and then there are strand J from a person without syndrome H. So looking into the diagram, we can see these nucleotides are the ones that are different. C is replaced with a T. Strand K shows a mutation in the DNA which has caused syndrome H. The enzyme IDUA helps to break down a carbohydrate in the human body. The enzyme IDUA produced from strand K will not work. Explain how the mutation could cause the enzyme not to work. So the process of the understanding would be the DNA codes for the amino acids. And the amino acids basically forms the polypeptide, which are which forms a protein which has an active site. So this is the enzyme. Enzyme is the protein, and this is the active site, and this is the substrate that fits the active site. So the mutation will cause the active site to change its shape, and as a result, the substrate will not be able to fit. Now, the answer can be written in this way. The mutation has changed one base of DNA from C to T. We will say the mutation has changed one base of DNA from C to T. This changes the amino acid coded or in the sequence, and this could change the shape of the protein formed. So the enzyme's active site was different shape. The enzyme no longer complementary to the substrate, or we can say the carbohydrate. A recessive allele causes syndrome H. A heterozygous woman and homozygous recessive man want to have a child. The question says, draw a Punnett square diagram to determine the probability of the child having the syndrome H. Identify any children with syndrome H. Use the symbols capital A and small a. So first of all, we need to understand what does the word recessive means here. In case of recessive, a allele is only expressed in phenotype if it is homozygous. So must be homozygous to present from both parent, alright, it must be received to show the effect in phenotype. So present in both chromosome can also be said inherited from both parents. So the woman is heterozygous. So the woman will be capital A with a small. The man is homozygous recessive. So the man will be small a and small a. So from the woman's part, these are the gametes the woman is going to pass on, and from the man's part, this is the gamete the man is going to pass on. From the woman's part, 50% of the child will receive dominant character and 50% of the child will receive homozygous recessive character. So we can say that this two child will have syndrome H. The probability is 2/4 or 0.5 or 50%. Food security is when a population has enough food to stay healthy. Lack of food security is a global problem. One way to maintain food security is to increase the efficiency of food production. Figure 10 shows how some pigs are farmed using intensive methods. Some people think the farming methods shown in Figure 10 are unethical. Suggest two other possible disadvantages of intensive farming. If pigs are kept in this particular condition, obviously the farmer might need to heat or cool down the pens. So heating requires use of fossil fuel. In some country, it might be cooling as well. And the animals are kept very close together. So there is a chance of spreading disease. So the answers that we can write in terms of the suggestion, the diseases can spread more rapidly. Heating will require an increased use of fossil fuel, or overuse of antibiotics could cause resistance. Explain how intensive farming of pigs increases the efficiency of food production. In order to score a level 2 for this particular answer, we must provide clear statement, which must be linked. Plus, we must give relevant explanation. So the answer can be written as follows. The pigs are kept in temperature-controlled environment, so less energy is required to maintain body temperature and less energy transferred to the environment. Their movement is restricted, so they are kept in cages. Therefore, less energy is required for movement and more energy is available for growth. A newspaper reported that food security is a serious problem in remote communities in Canada. This is because Aboriginal communities are eating fewer traditional foods. One traditional food eaten by Aboriginal communities in Canada is seal. Table 4. In 2004, the number of seals caught in thousands, 362, and in 2007 and in 2010, it is 67,000. Calculate the percentage decrease in the number of seals caught from 2004 to 2010. So, to find out the percentage change, we must find the final minus the initial divided by the initial times 100. So, in this case, the final is 67. The initial is 362 divided by the initial, which is 362 times 100, gives us negative 81.49. Now, the negative sign is in front because it is due to decrease. But when we are going to write down the decrease in seals, because the decrease is already mentioned, so we will only write 81.4. Because the answer says exam answer is 3 significant figures, so we will write 81.5. The conclusion in the newspaper might not be correct. Such as 2 reasons why. One of the reasons we can say is that they may eat other foods which are now available to them. So it is unclear if they are actually food insecure. And the other point that we can write is that we do not know if other traditional food have declined as well. Homeostasis controls the internal condition of the body. So how does the homeostasis in the case of insulin work? Whenever normal condition stays and the value of glucose concentration increases above the normal, then insulin is released, which then drives the glucose concentration down, and then the concentration of glucose returns back to normal. If the glucose concentration decreases, then the body releases glucagon. The pancreas releases glucagon, which then increases the glucose concentration and brings it back to normal. Explain why blood glucose levels are controlled in the body of someone who does not have diabetes. Explain how blood glucose levels are controlled in the body of someone who does not have diabetes. So we will say if the blood glucose concentration is too high, insulin is released from the pancreas. Insulin causes blood glucose to enter the cells and blood glucose then returns back to normal. If blood glucose concentration goes too low, glucagon is released from the pancreas. Glucagon causes the glycogen storage of the liver to be broken down into glucose and then released into the blood. Blood glucose concentration increases, returns back to normal. Compare how each type of diabetes is caused. Suggest how each type of diabetes can be treated. Now, when talking about diabetes, there are 2 types of diabetes. So, the first one is the type 1. In type 1 diabetes, no insulin is produced. Then there is type 2 diabetes. The body becomes insensitive to insulin due to genetic and environmental factors. Since the question is asking us to do comparison, so we'll have to use comparative language. So we will say, in type 1 diabetes, not enough insulin is produced, whereas in type 2 diabetes, the cells do not respond to insulin. And in the second comparison, we can say, in type 1 diabetes, it is treated with insulin injections, and in type 2 diabetes, it is treated with diet and exercise, plus some amount of insulin injections. Look at Table 5. Population of UK in 2015. Number of people diagnosed with diabetes. We can see 3.45 million people is diagnosed with diabetes. Estimated number of people with the undiagnosed diabetes. 0.549 million. Calculate the percentage of the UK population estimated to have diabetes. You should include both diagnosed and undiagnosed people in your calculation. So putting diagnosed and undiagnosed people together first. So total estimated people with diabetes, 3.45 times 10 to the power of 6 plus 5.49 times 10 to the power of 5. This gives us 3.999 times 10 to the power of 6. Then to calculate percentage of UK population, percentage from UK population, so that are estimated to have diabetes, we will take the number 3.999 times 10 to the power of 6. These are the people that may have and confirmed have diabetes to put together, divided by 6.5 times 10 to the power of 7 times 100. This gives us a percentage of 6.15, which is equivalent to two significant figures 6.2. So, estimated percentage of population with diabetes is 6.2. A urine test can be used to check for the presence of glucose in the urine. Diabetes can also be diagnosed with a blood test to measure the concentration of blood glucose. Suggest why a blood test is more reliable than a urine test. One of the main discussion point here that we can say is that there could be other reasons for which glucose can be in the urine, maybe other diseases. Then the second point we can say, or blood test gives the current or immediate glucose level, but urine might be, it can be behind like several hours because urine takes quite a long amount of time for it to accumulate and then be expelled out of the body. And there will not always be glucose in the urine. So any one of the 3 points would be correct. A blood test called the glucose tolerance test checks how well the body processes glucose. Concentrations of glucose in the blood are measured before and after drinking a glucose drink. Patients are not allowed to eat food for 8 hours before the glucose tolerance test. Suggest why patients are not allowed to eat for 8 hours before the test. One of the points we can write for this one is because the results are not affected by the glucose from the previous food that the person has taken. Or we can say 8 hours is sufficient time for insulin to have acted on any glucose that they have eaten from food. Or we can say so that there is a low starting point to show that additional glucose has an effect. Figure 11 shows the results of glucose tolerance test for two patients A and B. We can see patient A glucose concentration is above 7 in the beginning when the glucose drink is taken. And then patient A's glucose concentration rises and peaks at a much higher time compared to that of patient B, whose glucose concentration was much lower at the start, and when the glucose drink is taken, takes only half an hour to peak and then decreases back to normal again. Which patient has diabetes? Justify your answer. So definitely patient A, and the reason would be the glucose level are much higher than patient B, and then it remains high and does not fall to normal level. So patient A has a higher glucose concentration and then it doesn't fall. Whereas in patient B, insulin acts to lower the blood glucose concentration. So our answer will be patient A, and the justification will be patient A's blood glucose level are much higher and remain higher for longer. Endocrine glands produces hormones. Hyperthyroidism is caused by an overactive thyroid gland. Suggest what would happen in the body of a person with hyperthyroidism. So let's see what is the problem the person is facing, which is thyroid. Okay, what does it do? Thyroid produces thyroxine, which controls metabolic rate. The question says, what happens in the body? So there would be some physiological processes that will suffer. Example of this would be weight loss, muscle loss, digestion problem, palpitation of heart. So we'll write down the increased production of the thyroxine will occur due to the overactive thyroid gland. Therefore, the body's BMR would increase and rate of respiration would also increase. So when we are referring to BMR, we are talking about the basal metabolic rate, the metabolism rate when a particular person is resting. And other than that, we can also say there will be increased metabolic processes. Example, there will be higher rate of protein formation or breakdown as well. Describe the roles of FSH and LH in the menstrual cycle. FSH stands for follicle stimulating hormone. So it causes the eggs to mature and stimulate the ovaries to produce estrogen. Whereas LH, the luteinizing hormone, stimulates the egg to be released during ovulation. The combined pill is a contraceptive that contains progesterone and estrogen. The mini pill is a contraceptive that only contains the progesterone hormone. Has to be taken at the same time each day to prevent pregnancy. So in order to understand about the effect of this progesterone, we will have to understand the effect of FSH and LH properly. Let's see the question. The success rate of mini pill is preventing pregnancy is lower than that of the combined pill. Explain why missing a dose of mini pill would reduce the success rate of the mini pill. So let's see how progesterone is acting on FSH and LH, because the main job of progesterone is inhibiting FSH and LH. One of the main reason it has to be taken at the same time every day so that the same concentration is maintained in the bloodstream. So, first of all, we'll talk about FSH. FSH is released and produced by the pituitary gland. It causes eggs to mature and stimulates the production of estrogen. The other hormone is the LH. It is also produced in pituitary, and LH results in the release of egg or ovulation. So, as you can see, the action of the progesterone is very fundamental. Since progesterone can inhibit FSH and LH, then if there is no FSH, then there will be no follicle that will be matured. And if there is no LH, then there will not be any ovulation. So we can say missing a dose of the mini pill causes a drop in the progesterone level. So FSH is no longer inhibited, and a drop means LH is also no longer inhibited. Consequently, an egg may be matured and might get released due to the increase in LH. That's the end of this particular question paper, guys. Thank you for watching this particular video. Guys, question paper like this will be uploaded regularly. And we are doing a 365 days challenge where we will be uploading 365 question paper within a course of 1 year. The question paper will be mainly related to chemistry, biology, physics, and combined science question papers, starting from your GCSEs and then to your A-levels and then IGCSEs and international A-levels. So guys, subscribe to the channel and stay tuned so that you get your desired question paper. And I will provide a list of the question papers that I'm going to upload, and they will be in sequential order in terms of day. So one question paper is considered as a 1 day video. However, they are not in the same chronological order as the number of days. So we may be done with the uploading of the 365 question paper before the year ends, or it may take longer depending on how we can process them. So guys, stay tuned with the channel and support the channel. Let it grow. All right, guys. See you in the next video. Bye-bye.